microbiology textbook covers bacteria within the class Smallacutes. These are the smallest self-replicating organisms that were once thought to be viruses. Members of the class Smallacutes are more commonly referred to as either mollicutes or, probably the most common, Mycoplasma. There are many species identified in both plants and animals, but there are a few that are significant in humans, and at least 16 species have been isolated specifically from humans. There are two genera that are significant for our purposes in the family Mycoplasmataceae, and those genera are Mycoplasma and Ureaplasma, which we'll be discussing further in this chapter. The significant human pathogens we'll be focusing on are Mycoplasma pneumonia, which causes respiratory disease, and Mycoplasma hominis and Ureaplasma urolithicum that are associated with urogenital tract diseases. The textbook also mentions the genus Mycoplasma laidlawii in the family Mycoplasmaceae, and that single genus that it contains is the most significant human pathogen, but we'll not discuss this one in as much detail as Mycoplasma and Ureaplasma. So Mycoplasma, as was mentioned, are found on both plants and animals in a variety of different environments. The genus Mycoplasma, along with Ureaplasma, has been isolated from humans and cattle. Other genus have been found in various ecosystems, including soil, grass, plants, and water. One characteristic that the pathogens in the class Smallacute have in common, and this includes Mycoplasma, Ureaplasma, and Mycoplasma, are that they lack a cell wall, so they're cell wall deficient. Because of this, they're pleomorphic, and this also prevents staining by gram stain. In addition, the lack of the cell wall also makes them resistant to antibiotics that act against the cell wall, such as penicillins and cephalosporins. So the majority of beta-lactam antibiotics will not work against Mycoplasma, Ureaplasma, or Mycoplasma. As was mentioned, since Mycoplasma do not have a cell wall, they are pleomorphic in shape, so they can appear coxoid to tapered rods, and again, they cannot gram stain. Sometimes they're referred to as cell wall deficient or CWD organisms because of the lack of the cell wall. They're resistant to those cell wall active antibiotics, penicillins, and cephalosporins. These organisms are also slow growing. Generally, Mycoplasma require complex media for growth, with the exception of some of the species, such as Mycoplasma pneumonia or hominis, that can grow a little bit faster. Because they're fastidious facultative anaerobes, the enrichment they require in media usually involves cholesterol and fatty acids. And many times these specific conditions are found in cultures that have eukaryotic cells, so such as cell cultures. Mycoplasma are sometimes referred to as pleuropneumonia-like organisms, or PPLO, or eaten agent. So this name was after the person who discovered the organism. So you may see Mycoplasma referred to with these acronyms or names in certain studies. When Mycoplasmas grow in culture, they produce small colonies and often grow embedded beneath the surface of solid media. Some species form colonies with slightly raised centers that give them a classic fried egg appearance. Of note, we have used the description of fried egg to describe other organisms such as Yersinia, but you would be able to differentiate Yersinia from Mycoplasma based on the gram stain, because Yersinia would stain as a gram-negative rod, whereas Mycoplasma is pleomorphic and unable to be stained by gram stain. Mycoplasmas are transmitted via various routes, because as we saw, it can either present in respiratory infections or in urogenital infections. So sexual contact during delivery respiratory secretions, or fomites are possible modes of transmission. But it's important to remember that mycoplasma are very susceptible to heat and drying conditions, so appropriate transport media needs to be used. This figure shows mycoplasma appearing as a fried egg, and it's observed under a dissecting microscope to look at the colony morphology. Mycoplasmas adhere to the epithelium of mucosal surfaces in the respiratory and urogenital tracts, and they're not eliminated by mucus secretions or urine flow. As we saw, Mycoplasma pneumonia causes respiratory illness, so it's commonly found in the oropharynx, but when it is found, it is a cause of disease. Mycoplasma hominis is not typically found in the oropharynx, but it can be found in the urogenital tract, usually in about 10 to 50% of women. These figures show an example of the effects of Mycoplasma pneumonia on ciliated tracheal cells. So on the right is an example of an uninfected animal model's cells. And on the left is an infected animal model. So you can see the damage that's done to the ciliated tracheal cells. So of the various Mycoplasma species indigenous to humans, we'll be focusing on Mycoplasma pneumonia, hominis, and then Ureaplasma. So here you can see Mycoplasma pneumonia, its usual habitat or where it can be isolated is the oropharynx, and that's a common location of which it's isolated, but it's important to note that Mycoplasma pneumonia is not a normal commensal, and its isolation is always significant and it's considered pathogenic, whereas Mycoplasma hominis can be found rarely in the oropharynx, commonly in the urogenital tract, and it's commonly reported and is not necessarily indicative of disease. And this is the same for Ureaplasma. So the first mycoplasma we'll focus on is Mycoplasma pneumonia. So this organism may cause bronchitis, pharyngitis, or the common respiratory infection known as primary atypical pneumonia, also called walking pneumonia. It's important to remember with this diagnosis, atypical pneumonia, there's other organisms that can cause this, including chlamydophila pneumonia, Legionella. So the reason it's categorized as atypical pneumonia is because it has a different presentation than the pneumonia caused by Streptococcus pneumonia. The course of infection with Mycoplasma pneumonia, along with Legionella or chlamydophila, may also be mild, or typically may be mild, and it occurs more often in young adults. So again, it's presenting differently than strep pneumonia, which usually occurs in elderly people or with those people with underlying illnesses. And strep pneumonia also has a productive cough, which produces a sputum that's usually blood-tinged or rust in color, whereas these atypical pneumonias usually have a dry, unproductive cough. Again, as was mentioned, Mycoplasma pneumonia does not occur as a normal commensal organism, so its isolation is always significant and pathogenic. Mycoplasma pneumonia causes approximately 20% of reported pneumonias in the general population and up to 50% in confined populations, such as in military settings or prisons. School-age children and young adults are especially susceptible to infection. Although Mycoplasma pneumonia infection is not considered seasonal, more cases often occur in the fall or early winter. So outbreaks have been noted when teenage children return to school in the fall. So transmission is probably through aerosol droplet spray produced while coughing or via close contact. Many infections are actually completely asymptomatic, so that's about 90% of infected individuals, or they can be very mild, the symptoms. The most common presentation is tracheobronchitis, accompanied by pharyngitis. The incubation period is usually 2 to 3 weeks, and early symptoms are nonspecific, and they consist of headache, low-grade fever, malaise, and anorexia. Dry cough and earache are also accompanying symptoms. Extrapulmonary complications, those outside of respiratory involvement, can occur, but these are rare, and the complications include cardiovascular, central nervous system, dermatologic, and gastrointestinal problems. This chest radiograph is an example of what could be observed in atypical pneumonia. So there's usually nonspecific interstitial pneumonia and patchy infiltrates, and that's shown by a feathery outline. So usually a chest x-ray for a normal healthy individual should be clear, but here you can see it's a little opaque and that feathery outline can be observed. So typically you would just see the ribs and the black background behind the rib cage and the chest. So next we'll be looking at the two species of mollicutes that are associated with urogenital infections and then also can be associated with newborn disease. So although mollicutes do not cause vaginitis, both Mycoplasma hominis and Ureaplasma are associated with infections of the urogenital tract, and this may in effect play a role in vaginitis. So this table just summarizes Mycoplasma hominis and Ureaplasma specifically, and the diseases they cause. So here you can see the non-gonococcal urethritis has a strong association with Ureaplasma, whereas PID and pyelonephritis have strong associations with Mycoplasma hominis. And then we move into some of the newborn diseases that are associated. So low birth weight, infections such as sepsis, pneumonia, meningitis, these are strongly associated with Ureaplasma presence in the urogenital tract. Mycoplasma hominis is found in the lower genitourinary tracts of approximately 50% of healthy adults, but as seen in the chart, it has not been reported as a cause of non-gonococcal urethritis. It may invade the upper genitourinary tract and cause various presentations such as salpingitis, which is inflammation of the fallopian tubes, pyelonephritis, infection of the kidney and ducts, pelvic inflammatory disease, which could lead to sterility, and postpartum fevers. Mycoplasma hominis is frequently isolated from asymptomatic sexually active individuals. Because Mycoplasma hominis is an opportunistic pathogen, the immune status of the host is an important factor in the occurrence and severity of disease. Both Ureaplasma and Mycoplasma hominis can be transmitted to neonates during delivery. Since they're both involved with infections of the urogenital tract, and they can be normal flora of the lower urinary tract of women, they've been involved in both infections in urinary tract of newborns, along with significant infections of the fetus, such as chorioamnionitis, congenital pneumonia, chronic lung disease in premature infants, and meningitis of newborns with negative cultures. A couple other Mycoplasma species to note are Mycoplasma genitalium and Mycoplasma fermentans. So Mycoplasma genitalium, as the name implies, has association with urogenital infections. So it's been involved in non-gonococcal urethritis, cervicitis, endometriosis, and pelvic inflammatory disease. There is evidence linking Mycoplasma genitalium to some cases of tubal sterility. Mycoplasma genitalium is one of the species that's very difficult to culture, and it can take 2 to 3 months to grow and culture. In addition, the presence of this organism interferes with serology for other mycoplasma. More recently, with the use of polymerase chain reaction, or PCR, it's been found more frequently in urethral samples of men specifically with acute non-gonococcal urethritis. Mycoplasma fermentans is one species that's likely an opportunistic respiratory pathogen. It's not known how often it occurs in the respiratory tract of healthy individuals and children, but it's been detected in throats of patients with lower respiratory tract infection. Other groups of patients of which Mycoplasma fermentans has been recovered include patients with either respiratory illness or AIDS. Mycoplasma fermentans has also been documented as being isolated from patients' synovial fluid who have rheumatoid arthritis. As was mentioned previously, recovery of Mycoplasma species in culture is difficult. Isolation of Mycoplasma pneumonia from respiratory sites is not frequently attempted because growth can take several weeks and technical expertise is necessary. Mycoplasma hominis and Ureaplasma are less stringent in their growth requirements, but they still require cholesterol for synthesis of plasma membranes. Mycoplasma hominis is the only species that will grow readily on sheep blood agar and chocolate agars. Since Mycoplasma pneumonia is not as readily culturable, typically serology testing is used to identify the presence of the organism. Traditionally, with acute phase and convalescent samples collected 2 to 3 weeks apart, they'll demonstrate a 4-fold rise in titer. Since many of the early symptoms of Mycoplasma pneumonia infection are nonspecific, a thorough understanding of the disease process is necessary for interpretation of serum and culture results. So here you can see in this table, it shows the diagnostic manifestations of Mycoplasma pneumonia infection. So initially, at 5 days, symptoms are nonspecific and they can present as a cold or mild flu-like, and serology tests remain mostly negative after 10 days even. The symptoms persist and then serology results start to turn positive after 10 days. So here you can see at 10 days, IgM will begin to turn positive, and towards 15 days onward, then IgM and IgE that are specific for Mycoplasma will become positive. Specimens for Mycoplasma culture include body fluids such as blood, sputum, synovial fluid, cerebrospinal fluid, amniotic fluid, and urine, as well as wound aspirates and nasopharyngeal, cervical, and vaginal swabs. Tissue samples may also be submitted for culture. But remember, because of the lack of the cell wall, mycoplasmas are extremely sensitive to drying and heat. So ideally the specimen should be inoculated at the bedside, and if this isn't possible, they should be delivered immediately to the lab in transport media, such as SP4 or Shepherd 10B broth or 2SP, which are designed specifically for mycoplasma. Cotton-tipped swabs and wooden shafts should not be used because of possible inhibitory effects. So swabs should be made of Dacron, polyester, or calcium alginate with aluminum or plastic shafts, and the swab should be removed when the sample is placed in a transport media. Once the sample arrives in the lab, it should be frozen at -70 degrees if plating within 24 hours isn't possible. Because mycoplasma lack a cell wall, they're not able to be visualized by gram stain, so DNA fluorescence stains can be used. But it's important to note that these are not specific for mycoplasma. The stain that can be typically used is acridine orange, and it could be used as a screening test. Other methods have been developed or used, but they're not consistently accurate, such as antigen detection assays or PCR. With molecular testing for mycoplasma, results may be difficult to interpret if results are positive because patients with mycoplasma pneumonia infections can persistently harbor the organism for various lengths of time after their acute infection. Several culture media have been developed for the recovery of Mycoplasma and Ureaplasma, but no single medium is suitable for all species isolated from humans. Penicillin has been utilized to minimize bacterial contamination. With Mycoplasma hominis and Ureaplasma species being more rapid growers and relatively easy to recover, they've been cultured using SP4 broth and agar. Although Mycoplasma pneumonia is difficult to culture or not commonly done, the SP4 broth and agar is ideal for this organism, whereas the addition of arginine is needed for Mycoplasma hominis to grow, and the addition of urea is required for Ureaplasma species to grow. Once media is inoculated, broth media should be placed at 37 degrees Celsius under atmospheric conditions, whereas solid agar media may be incubated in an environment of room temperature, enhanced with 5 to 10% carbon dioxide, or in an anaerobic atmosphere of 95% nitrogen gas with 5% CO2. Mycoplasma hominis and Ureaplasma species colonies may appear within 2 to 4 days, whereas Mycoplasma pneumonia colonies may take 21 days or longer. As mentioned before, Mycoplasma pneumonia is generally not cultured, so it's not common to have a culture requested because it takes too long and the sensitivity is low. So typically, serology is used, evaluating 2 to 4 weeks apart for a 4-fold rise in titer. Mycoplasma hominis and Ureaplasma urolyticum can be cultured in liquid media, and then a pH change is needed before plating to an agar medium. So the liquid media used, as mentioned, are SP4, but also Shepherd's 10B or 2SP broth can be used. Once the pH change is observed, then the liquid media can be plated to enrich culture media and observed for characteristic colonies, having that fried egg appearance embedded in the agar. When checking for culture growth, Mycoplasma-like colonies can be stained with Dean's or methylene blue stain. This staining is performed by placing a small block of the agar on a glass slide, covering the colony with the stain, adding a cover slip, and examining the agar microscopically under low power using a dissecting microscope. Mycoplasma hominis again has typical fried egg appearance, with the periphery staining a light blue and the center dark blue, as seen in the figure on the right. Although it's not entirely conclusive, the growth rate, body site recovered from, and colony appearance can aid in the identification of mycoplasma. Glucose utilization in SP4 broth will cause an acid shift producing a yellow color, whereas arginine metabolism will produce a rise in pH, changing the indicator to a deeper red color. The change in color of the medium when it turns pink, because it requires arginine, is associated with Mycoplasma hominis. Ureaplasma species were once called T-strain Mycoplasma. The T stood for tiny, because their form are extremely small colonies that are difficult to see with the naked eye. Ureaplasma cultures on solid media should always be examined with a dissecting microscope. Urease activity of ureaplasma may be detected on solid agar containing urea and manganese chloride. This is in the U9B urease color test medium. Urease positive colonies are a dark gold and brown color because of the deposits of manganese dioxide. Similar to Mycoplasma hominis growing in culture, Ureaplasma creates a more alkaline media, which results in a pink color change in the media. Production of an alkaline reaction in 10B broth after overnight incubation of a urogenital specimen is suggestive of Ureaplasma urolyticum, whereas the alkaline shift in media with arginine within 24 to 72 hours is likely caused by Mycoplasma hominis. Identification of Mycoplasma and Ureaplasma is often done by typing methods using monoclonal antibodies and immunofluorescence. In addition, observation of plate media by using a dissecting microscope can be used, but of course this requires technical expertise. A direct plate immunofluorescent method that can be used involves fluorescent labeled anti-Mycoplasma pneumonia antibody flooded onto colonies on the plate. The plate is then washed and examined for immunofluorescence. The Chen assay is another method in which a fluorochrome is used to identify Mycoplasma-infected cells. It uses a DNA fluorochrome stain which highlights Mycoplasma species as small ovoid bodies distributed throughout a glacial acetic acid-fixed cell culture. In this figure, you can see example of mixed isolation of molecules. So the smaller colony is Ureaplasma urolyticum, or the tiny strain, or T strain Mycoplasma, whereas the larger colonies are Mycoplasma hominis. And you can see they have that distinct fried egg appearance. These figures show vero cells, which are monkey kidney cell lines that are artificially infected with Mycoplasma orale in this figure A, and Mycoplasma salivarium in figure C. Figure B is a negative control. This shows uninfected cells. As you can see, the Mycoplasma appear as small pinpoint fluorescent bodies throughout the background, and the different species of Mycoplasma have a different pattern formation in these fluorochrome stains. Table 25.6 in your textbook provides a good summary of the laboratory identification methods for the various Mycoplasma species of clinical significance and Ureaplasma. So as you can see, for Mycoplasma pneumonia, culture is not typically done because it's traditionally difficult, whereas with Mycoplasma hominis and Ureaplasma, culture is the method of choice. As was mentioned previously, indirect immunofluorescence and PCR testing assays are available, but these are being evaluated for all species, and some are used in research settings, and they're not commonly used for Mycoplasma hominis or Ureaplasma. Serologic testing is the traditional assay for Mycoplasma pneumonia, with enzyme immunoassay being the method of choice to detect reactive IgM, IgG, and IgA. As we already know, because they lack a cell wall, molecules are inherently resistant to beta-lactams. So those are the antibiotics such as penicillins and cephalosporins that act against the cell wall. In addition, they're resistant to sulfonamides, trimethoprim, and rifampin. CLSI has established standard minimum inhibitory concentration methods using broth microdilution and agar dilution. The broth microdilution is the most commonly used, whereas agar dilution is considered as the reference method, but this is a technically difficult procedure. In regards to Mycoplasma pneumonia, usually published susceptibilities are reported because the organism has a predictable pattern. Mycoplasma pneumonia is usually susceptible to tetracyclines, newer fluoroquinolones, and macrolides, although there has been reports of resistance to high-level macrolides, so this may alter the need for susceptibility testing. In addition, remember that Mycoplasma pneumonia being isolated is of clinical importance and indicative of infection, which may be symptomatic or asymptomatic, but Mycoplasma hominis and Ureaplasma urolithicum need to be evaluated if they need to be treated, because these are sometimes organisms that colonize the urogenital tract of humans. So again, as was just mentioned, when interpreting laboratory results in regards to Mycoplasma, when Mycoplasma pneumonia is detected by any method from a pulmonary or non-pulmonary specimen, it should be considered significant and a pathogen, whereas Mycoplasma hominis interpretation is not as obvious. So isolation from sterile sites is considered significant, but isolation from other sites requires differentiation from colonization. A detailed clinical analysis and the potential for repeat cultures is needed when determining whether Mycoplasma hominis is an infection or colonization. Of the mollicutes, Ureaplasma urolithicum is the most difficult to assess clinically. In urogenital specimens, it's been reported to colonize up to 70% of men and 45% of women with no apparent infection, so its isolation is not indicative of pathogenicity. Education of primary care providers is needed, usually by the laboratory, by indicating some sort of statement with culture results suggesting the potential for colonization versus pathogenicity. In these types of specimens, when isolating ureaplasma, quantification is also important, particularly in sterile specimens as CSF isolates. It's reasonable to assume that the isolation is significant. Table 25.7 in your textbook provides a good summary of frequently isolated pathogens that cause respiratory illness in various age groups. So here you have the organisms frequently involved as 1 through 6, and the key indicates which organisms are involved. So we have organisms such as chlamydophilin pneumonia, Mycoplasma pneumonia, Mycoplasma hominis. 4 indicates viral organisms. 5 are other bacteria and fungi, and 6 are Streptococcus pneumonia. So since we're focusing on Mycoplasma in this chapter, here you can see Mycoplasma pneumonia, which is indicated by number 2, is not commonly seen in newborn respiratory infections, but you can see that it has been associated with grade school age and up. Whereas Mycoplasma hominis, indicated by the key number 3, is involved in newborn infections, where it's not involved in infections of age grade school and greater. So the last thing to discuss or revisit are the treatment options. So again, as we know, due to the lack of the cell wall, they're inherently resistant to beta-lactams, which include penicillins and cephalosporins, and they're also resistant to sulfonamides, trimethoprim, and rifampin. There have been documented cases of high-level macrolide resistance, so we do need to start reevaluating the need for antibiotic susceptibility testing, because again, as was mentioned, typically they use published guidelines to treat infections. So the drug of choice is tetracycline, but this is used primarily for adults due to side effects that could affect children. So the specific treatments available for Mycoplasma pneumonia include erythromycin and tetracycline, as we saw, but again, tetracycline should not be used in children, and with erythromycin there have been reports of resistance. For Mycoplasma hominis infection, clindamycin or lincomycin are used, and this species is resistant to erythromycin. And finally, Ureaplasma urolithicum is resistant to clindamycin or lincomycin, so erythromycin should be used.